Hello, my name is Ryan Mayu, and I'm an assistant professor of biochemistry at Memorial University of Newfoundland. In this lecture, I'll be discussing controlling mitochondrial reactive oxygen species production for cell signaling. I will start this lecture with an introduction that focuses on giving a background on how mitochondria combust carbon to make energy and reactive oxygen species. This will be followed by a brief discussion on oxygen and its radicals and the difference between oxidative eustress and oxidative distress, and how these two terms properly define the dichotomous nature of reactive oxygen species in biology. This will then be followed by a few slides on the signaling properties of reactive oxygen species, focusing specifically on hydrogen peroxide. I'll then start moving through the core of this lecture, which is understanding mitochondrial ROS release. In the first part of this discussion, I will go through some of the basics of how mitochondria produce reactive oxygen species, focusing on the 12 different ROS-forming sites associated with energy metabolism. After this, I'll go through controlling mitochondrial reactive oxygen species production, focusing on proton leaks, supercomplex assemblies, and redox switches. And then finally, I will discuss the implications of redox switches in controlling mitochondrial reactive oxygen species signaling. Mitochondria fulfill various essential functions in metabolism, which includes meeting cellular energy demands through ATP production. This is achieved via the combustion of carbon and the liberation of electrons, which are then used to produce ATP by oxidative phosphorylation. Mitochondria are also the most important source of cellular reactive oxygen species, namely superoxide and hydrogen peroxide, the proximal reactive oxygen species generated by mitochondria, and the most important oxyradicals formed by biological systems. Importantly, the formation of superoxide and hydrogen peroxide relies on the same electron transfer pathways that mitochondria use to make ATP. And although ROS are damaging at high levels, it is now appreciated that low-grade mitochondrial ROS production, specifically hydrogen peroxide, is vital for regulating various cellular functions. Mitochondrial ATP and reactive oxygen species production begins when disparate forms of carbon are converted into common metabolites, which then enter the Krebs cycle. Once in the Krebs cycle, this carbon is oxidized further, resulting in the generation of the electron carriers NADH and succinate. NADH and succinate are then oxidized by complexes 1 and 2, and the liberated electrons are ferried all the way down the chain to complex 4, reducing molecular oxygen to water. Notably, this thermodynamically favorable downhill transfer of these electrons through the respiratory complexes is coupled to the pumping of protons into the intermembrane space, forming a transmembrane electrochemical gradient of protons. Proton return through complex 5 of the chain drives the phosphorylation of ADP, forming ATP. And then once formed, ATP is exported into the cytoplasm to do useful work in the cell in exchange for another ADP molecule. What is also notable here as well is that the proton gradient can also be utilized for the selective uptake of various types of solutes. For example, phosphate carrier is required for the uptake of phosphate, also utilizes the proton motive force. Proton return can also bypass complex 5 or solute transporters completely, re-entering the matrix through ANT or uncoupling proteins, or UCPs, through a process called proton leak. It is also notable that carbon does not exclusively enter into the mitochondria and be for metabolism through the Krebs cycle. It can also be oxidized by selective dehydrogenases localized specifically in the mitochondrial inner membrane, where following oxidation, these electrons are donated directly to the ubiquinone pool, bypassing the Krebs cycle completely. And of course, electron movement through the respiratory chain is not perfectly coupled to ATP formation. At various points of the chain, electrons can prematurely react with oxygen to form superoxide or hydrogen peroxide. Traditionally, complexes 1 and 3 and now complex 2 are viewed as the most important sources of ROS. Reactive oxygen species is a broad term and is often used incorrectly in biology. It is important to distinguish which reactive oxygen species are being studied, since oxygen radicals can significantly differ in reactivity, frequency of formation, and the biological consequences of their presence. Biological systems, like mitochondria, form 3 main reactive oxygen species, and this is attributed to the unique chemical features of oxygen. Since molecular oxygen has 2 electrons with parallel spins in its outermost antibonding orbital, this means it can only accept 1 electron at a time. Therefore, during its reduction to water, several reactive intermediates are formed. The Lewis structures for these reactive intermediates can be seen here. They include superoxide, hydrogen peroxide, and hydroxyl radical. Superoxide is membrane impermeant and is also a weak oxidant and reductant, and reacts with very few biological molecules. One exception here is actually iron sulfur clusters, which it can react very rapidly with. On the other hand, hydrogen peroxide can react with cysteine residues, and then, of course, there's the dreaded hydroxyl radical, which is by far the most reactive oxygen radical formed by biological systems. I'm going to talk a little bit about oxidative stress and damage because it's very important in terms of reactive oxygen species production within a cell. Oxidative stress refers to when reactive oxygen species levels are high enough to overwhelm a biological system's ability to eliminate them or repair resulting oxidative damage. In this scenario, mitochondria are producing large amounts of reactive oxygen species, in this case superoxide and hydrogen peroxide, which can accumulate within the cell, resulting in cellular damage. Superoxide and hydrogen peroxide accumulation can also exacerbate ROS production by other mitochondria through a process called ROS-induced ROS release, or RIRR. Here, the ROS formed by mitochondria damage other mitochondria or feedback on their source of production, resulting in an augmented ROS production by these mitochondria and increasing cellular damage. Now, in the previous slide, I indicated that superoxide is a weak oxidant, and also hydrogen peroxide only reacts with cysteine residues on proteins. However, their accumulation can damage and disable critical enzymes involved in cellular processes. For instance, hydrogen peroxide can also react with iron to give rise to hydroxyl radical. This is initiated by the reaction of superoxide with iron sulfur clusters, disabling these iron sulfur clusters, releasing labile iron, expanding the labile iron pool. This can then react with hydrogen peroxide to form hydroxyl radical, enhancing cellular damage. We're going to talk a little bit now about mitochondrial hydrogen peroxide signaling. Now, as I mentioned, superoxide and hydrogen peroxide are the proximal ROS formed by mitochondria. However, it is hydrogen peroxide and not superoxide that is used in signaling. The reason for this is that superoxide disables iron sulfur containing proteins, which are vital for mitochondrial function. This can have disastrous consequences, as discussed in the previous slide. Superoxide also has a very short half-life due to its rapid dismutation by superoxide dismutase. This is in contrast to hydrogen peroxide, which lasts longer in solution, has the capacity to diffuse through membranes, specifically through proteins called aquaporins, and can have a profound impact on the redox buffering capacity to affect cell function through changing cell redox buffering capacity, or direct oxidation of protein cysteine thiols, is called redox signaling in this figure, where it can modulate either insulin release, stress signaling, or apoptosis. It is clear that oxidative stress is two-sided. It can be adaptive, but if produced in excess, can be damaging. Thus, a term like oxidative stress can now be seen as inappropriate since it does not really capture the dichotomy of reactive oxygen species in biology. In an effort to capture these two different functions for reactive oxygen species, new terms have been developed to explain both the physiological and pathological functions for hydrogen peroxide. Oxidative eustress refers to a physiologically meaningful oxidative challenge that promotes adaptation through the oxidation of cysteine switches or alteration in redox buffering capacity. Oxidative distress, on the other hand, can be viewed as an excessive oxidative challenge that is deleterious due to the depletion of antioxidant systems and the irreversible damage to cell structures. This reimagining of how we define the role of reactive oxygen species in biology allows us to define hydrogen peroxide as a legitimate secondary messenger. Oxidative eustress is associated with adaptive and secondary signaling responses associated with cell proliferation, differentiation, and bolstering antioxidant defense systems, whereas oxidative stress involves high hydrogen peroxide and cell damage. To take advantage of its signaling properties and avoid the induction of oxidative distress at the same time, oxygen-using cells have developed various systems to keep superoxide and hydrogen peroxide in check. Superoxide is dismutated very rapidly in the matrix of mitochondria, as can be indicated here in this diagram, generating hydrogen peroxide either within the matrix or in the intermembrane space. The hydrogen peroxide formed on either side of the membrane can have multiple fates. For example, let's consider the matrix. Within the matrix of mitochondria, three systems quench this hydrogen peroxide. The two main ones are the thioredoxin system, which uses peroxyredoxins to remove hydrogen peroxide, and the glutathione system, which converts two glutathione molecules in the presence of hydrogen peroxide and glutathione peroxidase to glutathione disulfide. The third system seems to be localized specifically within hepatocytes and has also been documented to occur within cardiomyocytes, and this is catalase, which dismutates hydrogen peroxide directly to water. Notably, the thyrodoxin and the glutathione systems also rely on the production of NADPH, which can be seen on the right of the diagram, which is linked to energy metabolism. There are some important considerations when considering hydrogen peroxide signaling, and for this I'll be discussing cyclic AMP, a very common secondary signaling molecule. The concentration of secondary signaling molecules like cyclic AMP is controlled through its production and its degradation. A similar concept can be applied to hydrogen peroxide, a powerful signaling molecule that can influence a range of cellular programs. Antioxidant defense systems are integral for controlling its concentration through degradation pathways. However, less is known about how mitochondria control hydrogen peroxide production. Indeed, similar to cyclic AMP, hydrogen peroxide levels must also be controlled through its production. So the first thing I'll be addressing is not controlling mitochondrial peroxide production per se, but rather the different sites of production. In this particular case, complex 1 and 3 are traditionally viewed as being the most important sources of ROS in various tissue types. However, recent evidence indicates that mitochondria contain up to 12 superoxide and hydrogen peroxide forming sites. So then the question becomes, which release site serves as the most important source of mitochondrial ROS in peroxide signaling? The second thing I will discuss is mitochondria and how they control hydrogen peroxide release from these different sites. What are the mechanisms are invoked to control hydrogen peroxide release from mitochondria? So the first consideration we will look at is simply understanding mitochondrial superoxide and hydrogen peroxide release. Over the past 15 years, it has become clear that mitochondria do not release superoxide and hydrogen peroxide exclusively from complex 1 and 3. It is now appreciated that mitochondria can contain up to 12 superoxide and hydrogen peroxide forming sites that are associated with carbon metabolism and respiration. This is indicated here by the red stars in this diagram. These sites typically use flavin groups, such as flavin mononucleotides or flavin adenine dinucleotides, or semiquinone radicals, to form either superoxide or hydrogen peroxide. The 12 sites can be broadly classified into two groups. The first group is the NADH/NAD+ isopotential group. These are enzymes that are associated with keto acid metabolism and are also associated with NADH production. Within this group, we have pyruvate dehydrogenase, 2-oxoglutarate dehydrogenase, 2-oxoadipate dehydrogenase, and the branched chain keto acid dehydrogenase. The second group donates electrons directly to the quinone pool, bypassing the Krebs cycle. These enzymes include your sulfide quinone oxidoreductase, dihydroorotate dehydrogenase, proline dehydrogenase, glycerol 3-phosphate dehydrogenase, ETFQO, which is involved in fatty acid metabolism, and complexes 1, 2, and 3. Most of the work associated with identifying the sites for superoxide and hydrogen peroxide production have been carried out by Dr. Martin Brand's group, with mitochondria isolated from rat skeletal muscle tissue. This can be seen here in the figure on the left. The information yielded from these studies has been very useful in identifying which sites serve as high capacity ones under different bioenergetic and physiological conditions. Again, focusing on the graph on the left, the highest capacity site for ROS production happens to be complex 3. And within the quinone isopotential group, other sites include complex 1, complex 2, and glycerol 3-phosphate dehydrogenase. However, when we focus on the NADH to NAD+ isopotential group, it is shown that when you're metabolizing keto acids like 2-oxoglutarate or pyruvate, that the 2-oxyglutarate dehydrogenase and pyruvate dehydrogenase produce 8 times and 4 times more reactive oxygen species than traditional sources like complex 1. These seminal findings were able to show that the major site for production can vary significantly depending on which type of substrate is being oxidized. In addition, this work has shown that the most important factors influencing ROS production are the concentration of the electron donating site, the redox state of the electron donating site, and the substrate type. Now I'm going to talk a little bit about forward and reverse electron flow and its implications in reactive oxygen species production by the electron transport chain. It is very important to emphasize here that ROS can be formed during forward and reverse electron flow through the chain. In the first example here, I'll just talk about forward flow from the Krebs cycle. In this example, oxidation of a carbon source that is associated with the Krebs cycle releases electrons, which are then translocated through the respiratory complexes, reducing oxygen at the end of the chain. And of course, along the way, electrons can leak out, as discussed in previous slides, to make reactive oxygen species. The oxidation and reduction of quinone, on the other hand, can yield reactive oxygen species via forward and reverse electron flow. I will use the example of succinate metabolism first. Succinate is oxidized by complex 2, and the electrons move in a forward sense up the chain all the way to complex 4, producing reactive oxygen species at the level of complex 2 and 3. However, these electrons can also move backwards to complex 1 and form superoxide or hydrogen peroxide. In fact, reverse electron flow from complex 2 to complex 1 is a potent source of reactive oxygen species in various organisms and tissues and has been associated with different pathologies like heart disease. It also turns out, based on work done by Dr. Martin Brand's group, that fatty acid oxidation can also flow forward to complex 3 or backwards to complex 1 and 2. It is also the same for various dehydrogenases that couple carbon oxidation directly to the reduction of the quinone pool. Using similar methods and different inhibitor substrate combinations, our group has demonstrated that liver mitochondria use different combinations of enzymes as high capacity sites for superoxide and hydrogen peroxide production. For example, in contrast to muscle, we have found that liver exclusively uses 2-oxyglutarate dehydrogenase and complex 3 as the main sources for reactive oxygen species. Pyruvate dehydrogenase makes up a small fraction, as well as other sites. In addition, our group is currently investigating the ROS release profiles of other tissues. For instance, our group has recently found that Krebs cycle-linked substrates, pyruvate and 2-oxyglutarate, are excellent sources for reactive oxygen species production in liver mitochondria, but not in cardiac mitochondria. By contrast, succinate produces 4-fold more ROS in cardiac mitochondria. So in previous slides, I discussed the importance of reverse electron transfer, or RET, in ROS production in mitochondria. Turns out that 2-oxyglutarate dehydrogenase and pyruvate dehydrogenase also have the capacity to form reactive oxygen species by reverse electron flow from NADH. Superoxide and hydrogen peroxide production by reverse electron flow to OGDH was documented more than a decade ago. However, few studies have focused on delineating its importance in physiology. Our group recently compared the ROS forming potential of purified 2-oxyglutarate dehydrogenase and pyruvate dehydrogenase of porcine heart origin and found that OGDH was a much higher capacity site for production during reverse electron transfer. It is important to point out that reactive oxygen species production by reverse electron flow can be stimulated by low levels of NADH, which can be depicted here in the graph on the right. The rate of production also increases with higher NADH concentrations. It was speculated more than a decade ago by two separate groups that reverse electron flow from NADH to 2-oxyglutarate dehydrogenase may contribute to the pathogenesis of neurological disorders. Under normal conditions, NADH is oxidized by complex 1, as can be shown here in this diagram, and the electrons flow forward, contributing to respiration. However, a blockage or a deficiency in complex 1 can result in the accumulation of NADH, enhancing electron backflow to OGDH and PDH. Our group has recently started looking into determining if NADH can contribute to ROS production by reducing reverse electron flow to OGDH and PDH in mitochondria. For this, we utilized complex 1 deficient mice and isolated the mitochondria from the liver and cardiac tissue. We were able to find that ROS production by these permeabilized mitochondria is NADH dependent, suggesting that ROS production by OGDH and PDH from reverse electron flow from NADH is physiologically relevant. So I'm just going to provide a quick overview of the many sources of mitochondrial reactive oxygen species. First, I covered how mitochondria can contain up to 12 sites for ROS production. Tissues display preferences for which superoxide hydrogen peroxide generating enzymes will serve as high capacity sites. This could be related to metabolic or nutrient preferences for a given cell type, or be associated with differences in how hydrogen peroxide superoxide production is regulated. Then finally, I talked about reverse electron flow from NADH to 2-oxyglutarate dehydrogenase and pyruvate dehydrogenase, and other keto acid metabolizing enzymes, which still needs further exploration. Further exploration is required because of the potential for it being physiologically relevant. NADH backflow could be a potent source of hydrogen peroxide in cell signaling, but can also be a potential source of oxidative distress if complex 1 is disabled.